Hi everybody, so today we're going to be looking at microglial cells. So let's go ahead and get started on this. And basically, as you see, this means small and glial means glue. These are glial cells, right? And glial cells, as we know, as we're starting to discover, these are cells that are in the central nervous system that are not nerves, but they help the central nervous system function, right? So let's look at the role of what these do. Basically, they're gonna clean up the CNS. So what I mean by that is they're going to eliminate any debris that might be in the central nervous system. They're also gonna go around looking to see if any synapses are old and need to be eliminated. And so they're gonna basically clean all that up, right? We'll go more into that in just a minute. Part of the cleanup process is what we call synaptic pruning. And what we mean by synaptic pruning is when you were born, you have a lot more synapses than you actually need. So as time goes on from birth until about puberty, what these microglial cells will do is they will eliminate all those excess synapses that are in the brain and spinal cord, right? So that they're gonna play that role there as part of the cleanup. And again, they'll get rid of old synapses too, or old nerves, or basically any other debris that's in there. But remember, these are also, like we said, they're part of the immune system, so these are a type of macrophage. And so because they're a type of macrophage, what they're also going to do is they're going to eliminate pathogens. Okay, so let's go ahead and take a look at what these do. Okay, we'll see how they function right now. So let's go ahead and get started on that. Okay, so now, this is what the microglial cells are going to do. Is let's say we have nothing going on, and this is going to be my microglial cell. This is an inactive one, right? This is inactive. It's going to have these little processes, these little arms that are reaching around, just trying to feel how everything is doing in the CNS. Okay, so let's say we have an old synapse we want to get to, right? We want to eliminate this synapse. So now, here's the synapse. And what's going to happen is the synapse is going to start to release some chemicals, right? It's going to release some things, inflammatory chemicals. And what's going to happen is these arms are going to sense that something's not right. So when it does that, this is my, like I said, this is my inactive microglial cell. This is going to become activated. So what's going to happen next is this is going to retract the arms and grow in size, right? So right now it's inactive, right? Now it's going to become active. This is going to be an active microglial cell. So here's my active microglial cell now, right? And then here is that synapse still. Okay, and then what this is going to do is, because of everything going on, this is going to release chemicals that are going to basically cause inflammation. It's going to secrete reactive oxygen species. It's going to secrete nitrous oxide. It's going to secrete free radicals. And it's going to secrete some cytokines. All right, so then once it secretes these, what's going to happen now to my synapse, and this could also be onto a pathogen, what's going to happen now is this is going to break apart. So now here's the parts of what was my synapse, right? And then what's going to happen is this is going to phagocytize these. So now it's going to phagocytize these, right? And it's going to bring this into the cell. So now it's inside the cell. We do it to all of these. I'm just drawing one right here because it's a lot easier to do. And now we have our debris in there, right? That's in there. So now what's going to happen is inside of here, we're going to have something that we're going to call a lysosome. And what the lysosome has in it is it has some digestive juices in there, right? And some enzymes. And what that's going to do is that's going to connect onto here. And then you guys should know this already. What's going to happen is it's going to cause this to break apart. So now, even though we've already broken it apart once, now we're breaking it apart into even smaller pieces. So now we have these small pieces here. Now, being that these are a type of macrophage, as you know what macrophage is, what's going to happen is they are antigen presenting cells. So what's going to happen now is this is going to come out here, and it's going to present some of this what's left over on its cell membrane. So then this is going to basically release, like we said, we have cytokines, and then we're going to have our bloodstream, right? And in the central nervous system, remember, we call this the blood-brain barrier. So I'm going to have my little tight junctions there. And what this is going to do when it releases those cytokines, it is going to cause T cells, T lymphocytes, are going to leave the blood-brain barrier, and they are going to come and they're going to start interacting with this. Now remember, we can't have antibodies come through here because of the fact that they can't get past the blood-brain barrier because they're too big. So now I have my T lymphocyte right here, right? And then what this is going to do is this is going to activate the microglia to secrete more of the cytokines and these other inflammatory chemicals to basically continue to attack this until it clears it up. Okay, now, it can do this just when these are getting old and they're no longer working properly, or it can also do this, like we said, in synaptic pruning. The same thing's gonna happen is, let's say we have a bacteria, right? If I had a bacteria in this area, it would do the same thing. It's going to release these, cause the breakup of these, take it in, present it, and then the T lymphocytes would also help destroy this, okay? So that's it for microglia. Thank you so much for watching. If you enjoyed the video, please hit the like and subscribe button, and we will catch you next time.